Hi students, today let us discuss about the process of transcription in prokaryotes. And before starting this part under transcription, I want to rectify two mistakes that were committed in the last class. One, the sequence of Tata box. The sequence of Tata box is about TA, TA and AA. This is a sequence. The Tata box has a nucleotide sequence that is about 6 nucleotides, TA, TA and then AA. And now TA, 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 what I mentioned. This is one of the corrections. You make it. Another one regarding the presence of mRNA, the monocystonic gene and polycystonic gene were suggested by two different people. One, Francois Jacob, another one, Jacques Monod. These were also responsible for the operon concept, lac operon concept. I mentioned in the last class, the monocystonic gene and polycystonic gene were reported by Richard J. Robert and Philip A. Shaw. So they were noted for split genes, not for this one. So this is another mistake. So you make a correction of it. mRNA, monocystonic gene, polycystonic gene, all were reported, suggested by Francois Jacob and Jacques Monod. Simply we can say Jacob and Monod and not Richard J. Robert, Philip A. Shaw, what I mentioned in the last class. Okay, these are the two corrections you have to make it. I am sorry for that one. What is the mechanism adapted by prokaryotes during the process of transcription? So normally we need an enzyme for the synthesis of RNA. The name of the enzyme is RNA polymerase. RNA polymerase. RNA polymerase. This is the enzyme responsible for the synthesis of what we have mRNA. Now in the case of eukaryotes, we have three different types of polymerase enzymes. But in prokaryotes, there is only one RNA polymerase. You know well about, there are three different types of RNAs. One, mRNA. Two, that is tRNA. And the third one, ribosomal RNA. So mRNA, tRNA and ribosomal RNA, these are the three types of RNAs. Now these three types of RNAs are synthesized by a single RNA polymerase enzyme, unlike the eukaryotes. In eukaryotes, there are three different types of RNA polymerase. Polymerase 1, 2 and 3. We'll discuss. For the synthesis of these three, what we have RNAs. Now the messenger RNA is acting as a template. This is acting as a template, model, carrying the message from the nucleus to the ribosome. Now the tRNA, it brings amino acid and reads the genetic code. During protein synthesis, it brings amino acid and reads the genetic code. This is the role of tRNA, brings amino acid to the ribosome by reading the genetic code. We'll discuss. Because in messenger RNA we have the message in the form of codon or code, and that message is being read by what is called tRNA to select the right type of amino acids from the cytoplasm. We'll talk about under the translation process. Now the ribosomal RNA, it is present in the ribosome. It plays a structural and catalytic role. Structural and catalytic role. That is during translation. So it plays a structural and catalytic role during translation process. So these are the three types of RNAs which are taking part in the process of protein synthesis. And all the three types of RNAs are synthesized by a single RNA polymerase enzyme. Unlike that's eukaryotes. So mRNA, the template, tRNA brings amino acid and reads the genetic code. Accordingly, only it selects the right type of amino acid from the cytoplasm. Now, the ribosomal RNA is formed in the ribosome. It forms a structural component of the ribosome as well as acting as a catalyst. So, structural and catalytic role when during the translation process, the next step in gene expression. Now, what is happening? Now, this RNA polymerase, there's normally three events. One, initiation process. During transcription, there are three events. One, initiation. Another one, the second event, elongation. The third event, just a termination. These are the three events: initiation, elongation, and then termination. Now, what will happen? The RNA polymerase normally utilizes the substrate in the form of nucleoside triphosphate and also the polymerases, and in a template dependent manner based on following the rules of complementarity. So, if you are taking what we call the RNA or mRNA, the bases present in mRNA are similar to the coding strand and also complementary to, that is what is called the template strand. So, following the rules of complementarity and also just you see that on a template dependent manner, the nucleotides are added one by one in a chain-like structure by what we have RNA polymerase. So, once you see that one as a polynucleotide chain is formed, normally how? By the addition of the nucleotides in the form of nucleoside triphosphate, which is acting as a substrate in a template dependent manner following the rules of, that is, complementarity. So, they are added one after another. Once the polymerase enzyme reaches what is called the terminator region, see, the structural gene is responsible for the transcription to form the RNA. Now, the polymerase enzyme is leading. So, the polymerase enzyme is leading towards what is called the termination point. And normally only a small stretch of RNA, the one which is newly synthesized, is bound with RNA polymerase. Not the entire RNA is bound with RNA polymerase. Only a small stretch of RNA, the one which is being formed continuously, is bound with the RNA polymerase. Now, this is what's called the growing RNA. It is found attached to the RNA polymerase, just only a small stretch. Now, you see that one, we have the starting point. The RNA polymerase cannot initiate the process of transcription. It needs the help of a factor. That factor is called sigma factor. Now, the sigma factor associates with the RNA polymerase transiently, temporarily, to initiate the process of transcription. So, the RNA polymerase initiates the process of transcription. Normally, the RNA polymerase binds to, the promoter region. So, the RNA polymerase binding site is nothing but the promoter, where you have the Tata box. That one directs the RNA polymerase to the site of transcription. And now, the sigma factor initiates the process of transcription. Once the process of transcription is initiated, the sigma factor is relieved from the RNA polymerase core enzyme. You see, it is left out. Okay, first the RNA polymerase binds to the promoter region. Okay, there we have the Tata box which directs there in the case of prokaryotes, that one is called Prignow box, which directs the RNA polymerase to the site of what is called transcription. But the transcription process is initiated by a factor which is associated with the RNA polymerase transiently, temporarily, and that is called sigma factor. Once transcription begins, initiated, now the sigma factor is left in the promoter region. So, the RNA polymerase binding site is called the promoter. Okay, now the addition of nucleotides taking place one after another. When the RNA polymerase reaches the termination point, there is a sequence in the DNA or some other protein. Now we have two types of termination. One type of termination what is called DNA dependent termination. In DNA dependent termination, what will happen? Now the newly synthesized RNA, what is called the nascent mRNA here, is being relieved from, that is DNA strand, with the help of the sequence present in the DNA cell. That is called DNA dependent transcription. But in the case of prokaryotes, we have a protein. Once the completed RNA reaches actually the termination point, there we have protein what is called the rho factor. This rho factor is called as the termination factor. So sigma factor is an initiation factor for the initiation of transcription, and rho factor is what is called the termination factor. Once transcription is completed, it causes a termination of transcription, so that what is happening, the nascent mRNA falls off from the DNA. When it is falling off, the RNA polymerase along with the rho factor is also falling off. That is the end. So normally I mentioned already, there is a bubble-like structure is formed while the two strands of DNA separated at the point of origin of what is called the transcription. That is called transcription bubble, this area. So this is a simple process. But we have three events: initiation by the sigma factor, elongation by what is called the RNA polymerase enzyme, and termination by what is called the rho factor. So initiation factor, that is sigma factor, elongation of the growing mRNA by means of what is called RNA polymerase enzyme, and in the case of termination, we have the termination factor which is called rho factor. Now, we have only single RNA polymerase. How does the single RNA polymerase do the activity of initiation, elongation and termination? That is one question. You know that one, the RNA polymerase has only the ability or the capacity of actually elongating the DNA, elongating the RNA by polymerization process. It cannot have the ability of initiating or just actually finishing or terminating the process of transcription to relieve the newly formed mRNA from the DNA. Now, the RNA polymerase associates, that is transiently, with the initiation factor called sigma factor and the termination factor called the rho factor, transiently, temporarily, either to initiate the process of what is called the transcription or to terminate the process of what is called actually transcription process. So sigma factor, initiation factor and then rho factor. Okay, so once the RNA associates with these factors, instruct the RNA polymerase either to initiate or to terminate the process of transcription. Now, some more points related to the transcription in prokaryotes. Now, in the case of prokaryotes, there is no processing. What do you mean by that one? We will see that what is processing. There is no processing in the case of prokaryotes, but we have processing in the case of eukaryotes. This is because of the split gene nature of the eukaryotes. So since there is no processing in the case of that is transcription in prokaryotes, to make or to become, that is what we have the RNA active. Again, since the transcription and translation processes taking place in the same compartment, meaning that there is no definite nucleus separated from the cytoplasm. That is called same compartment. The nucleoid and cytoplasm present in one compartment. Many times what will happen, the process of translation can begin much before the transcription is complete. The transcription is fully completed. The meaning for that one, transcription and translation occur simultaneously This is because we have both, that is the DNA and the cytoplasm present in the same compartment. So I mentioned about we have the transcription, translation occurs simultaneously. So this is due to, that is the presence of the genetic material, or we can say this is due to the genetic material which is not separated from the cytoplasm or cell organelles by the nuclear membrane. So we have the transcription and translation occurs what we have co-transcriptionally. That means transcription, or we can say translation occurs co-transcriptionally. Transcription and translation occurs simultaneously, that is coupled with each other because of what we have only one compartment. So the beginning of translation occurs many times just before the transcription process is completed. The reason for that one, there is no separation of genetic material from the cytoplasmic organelles. So the transcription is coupled with the translation. And also the translation occurs co-transcriptionally, that is both occurs simultaneously. I will repeat once again in a short. So first the RNA polymerase binds with the promoter region, thereto the pregnant box with the help of what is called the sigma factor. It is a factor which makes the RNA polymerase a coenzyme to bind with pregnant box. That is similar to the Tata box. And that one directs RNA polymerase to the site of what is called transcription. So the RNA polymerase binding site is nothing but the promoter, where only we have the printout box. Now the process of transcription begins. Once the process of transcription begins, now the sigma factor is no more required, it is left. You see, it is left. Now the RNA polymerase just causes the elongation of the growing polypeptide chain by adding the nucleotides one after another. Usually at the 3' carbon atom is the only process. Even the synthesis of RNA occurs only in 5' 3' direction. Either the transcription or we have the replication, both occurs only in 5' 3' direction. So when the RNA polymerase reaches the final stage, when the structural gene reaches what we have the termination point, where we have the terminator in the form of what is called rho protein. The rho protein causes the release of, that is the fully transcribed mRNA from the DNA. So the nascent mRNA falls off from the DNA, and so also the rho factor and RNA polymerase. Unlike prokaryotes, the process of transcription in eukaryotes is somewhat a complex nature or a complicated structure or complicated process. Here, again, unlike prokaryotes, there you have only one RNA polymerase, but here there are 3 RNA polymerases. With the division of labor, the meaning for that one, each one is responsible for the production of a specific type of RNA. Now RNA polymerase 1. It is concerned with the production of ribosomal RNA, the structural RNA making the ribosome. Now the units, normally that's ribosomal RNA consists of 28S, 18S and 5'S. So S refers to the Svedberg unit, the sedimentation coefficient. So these are all the different sizes of what is called ribosomal RNA, the units, synthesized all by RNA polymerase. Now RNA polymerase 2 synthesizes mRNA, that is what is called immature mRNA, unprocessed mRNA or precursor mRNA. And also we can say heterogeneous nuclear RNA, HN referring to heterogeneous nuclear RNA, heterogeneous. So the HN referring to heterogeneous nuclear. This is what we have HN, heterogeneous. What's the meaning for that one? It's being formed of just a coding sequence, non-coding sequence. Because in the case of eukaryotes, the gene is monocystronic. You know that one gene, one polypeptide and one protein. Again, it is called as a split gene. It is being formed of two different units. One, we have the exons. We will talk about later, the coding sequence. And then interrupted by what we call intervening sequences, introns, non-coding sequences without any message. And now, a heterogeneous RNA consists of coding sequences called exons and non-coding sequences introns. That's why it's called not homogeneous, it's called heterogeneous, formed of two different components, introns and exons. Okay, so anyway, the RNA polymerase II is responsible for the synthesis of heterogeneous RNA. This is called the precursor RNA or immature RNA. Then what we have later, the introns are being removed, the exons are joined together to form a mature RNA. We will discuss later. So the meaning for heterogeneous RNA, it is being formed of exons and introns, the coding sequences and non-coding sequences. That is being synthesized by what is called RNA polymerase II, and from it only we are getting what is called the mature mRNA. Now the RNA polymerase III, it is concerned with the production of the third RNA, tRNA. Then in what we have single-stranded nuclear RNA, single-stranded nuclear RNA or simple nuclear RNA, and also one of the units needed for the formation of ribosome, 5S ribosomal RNA. 5S ribosomal RNA. It is the one which is being added to the ribosome along with what is called 28S, 18S, 5.8S and also 5S. These are the different units that make up what we call the ribosomal RNA. So this is about the division of labor about the RNA polymerase in the case of eukaryotes. In eukaryotes, the functional structural gene is called actually monocystronic gene because it produces one mRNA which is translated to form a single protein. Then again, the structural monocystronic gene is called a split gene. That was reported by Richard J. Robert and then Philip A. Shaw, 1977. So this split gene property is absent in the case of prokaryotes. That's why they are not undergoing any processing. Once formed, transcription is completed, the mRNA is ready. But here, what will happen? The gene is what is called split gene. What's the meaning? It consists of two components. One is what we call interrupted coding sequences. They express the sequences. So the coding sequences are called the exons. The coding sequences are called exons, which are intervened by non-coding sequences, which are not expressed. They are called introns. As we have two components in the gene is called split gene, proposed by Robert and Shaw. Now what is happening during transcription? So this is in DNA. Both exons and introns are transcribed. That's what's happening. Both exons and introns are transcribed. But what is happening during translation? The introns are not translated. So the unit which is being transcribed and translated is exon. The unit which is transcribed but not translated is intron. Transcribed but not translated. So after transcription we have formed what is called the mRNA, as it contains both introns and exons. See, introns and exons. This mRNA is called heterogeneous mRNA. It is also called immature mRNA or unprocessed mRNA or precursor mRNA, as it contains both introns and exons. We don't want introns. They should be removed from this mRNA. For that, now the mRNA is undergoing a process that is called processing of mRNA. So in the case of prokaryotes, there is no processing. Now the processing is otherwise called splicing. What is splicing? The removal of introns. You see the introns are removed. The removed introns. And the joining of exons. You see all the exons are joined. The functional coding sequences are joined. And the non-functional non-coding sequences are removed. That process is called splicing or processing. But in the case of eukaryotes, they have to undergo additional process. What are the additional processing? One processing is called capping, addition of cap. Another processing is called tailing. What is capping? At the 5' end, there is an addition of a special type of nucleotide called, that is what is called guanosine, that is what is called guanosine methyl triphosphate. Guanosine methyl triphosphate. Actually it's called guanosine 7-methyl triphosphate. Guanosine 7-methyl triphosphate. It is added at the anterior end. This process of adding guanosine triphosphate, guanosine methyl triphosphate is called the capping process. It acts like a cap. What's this actually importance? It protects the mRNA from degradation, not being attacked by nucleases. And also, it is used for normally to be recognized by the ribosome. So this mRNA is to be recognized by what is called the ribosome. Further, the cap is needed. It is nothing but 5-methyl, that's actually 7-methyl guanosine triphosphate added to the 5' end. Then at the 3' end, that is called the tail region, addition of what is called adenine nucleotides. Addition of adenine nucleotides. Nearly 200 to 300 nucleotides are added, particularly adenosine nucleotides. Adenosine nucleotides are added, or we can say adenine nucleotides are added at the tail end. Nearly 200 to 300 actually nucleotides are added. That's why it's called polyadenine tail. Polyadenylate tail. Polyadenylate residues is being normally synthesized with the help of enzyme. What is its function? Again, protection. It protects the terminal end not being attacked by the nucleosis and provides stability to the mRNA. These are some of the functions of what we have, that is polyadenylate tail. So, one process what's called splicing. Another two processes, additional processing that occur in the case of that is eukaryotic transcription process. One is capping process, addition of a special type of nucleotide, guanosine methyl triphosphate, and also addition of many adenine nucleotide or adenylate nucleotide at the tail end, that is called polyadenylate tail. That process called capping. These are all the additional processes taking place in the case of eukaryotes to provide additional stability to the mRNA when compared to, that is, prokaryotes. So in the case of prokaryotes, there is no splicing process, there is no capping, there is no tailing. It is a simple organization. But in the case of eukaryotes, it's a complex organization. The process also very complex in nature. That's why we have additional processing also taking place. Okay, now we are getting what's called the mature mRNA. The mRNA contains only the exons, the cap and the tail.